Okay, good morning, everybody. All right, so today we are going to talk about the ketogenesis or biosynthesis of ketone bodies. And we already finished the fatty acids oxidation, right? So you guys know why we need to oxidize fatty acids, in which physiological conditions we need utilize the fatty acids. Which conditions? Fasting, right? Starvation. And we also mentioned that the ketone bodies and fatty acids are the main energy fuels during the starvation, right? Because we have a limited availability of glucose, and although we can break down the glycogen to release glucose, or we can produce the glucose through the gluconeogenesis, but still we need to spare glucose, for which organs, tissues are very important, need to use glucose. Brain and red blood cells, right? So because they heavily rely on the glucose for energy use. So now let's switch gears to the ketogenesis. Okay, why do we need ketone bodies? What is a ketone body? We just talked about it, right? We need ketone bodies. I just talked about that we can use fatty acids, right, for energy, but why we also need ketone bodies? Right. Thank you. So she said because brain cannot utilize fatty acids, right? Fatty acids, two molecules too large cannot, get to the brain through the blood-brain barrier. So ketone bodies are much smaller molecules, right? Not able to access the brain. So primarily because the brain cannot utilize fatty acids during the starvation, so we need to synthesize ketone bodies. And when do we need to synthesize ketone bodies? When? Starvation, right? Very clear. So starvation, particularly prolonged fasting, fasting for longer than 2 days, then our body start to synthesize ketone bodies. So this will be under the physiological conditions, right? And there are also another condition that ketone bodies also being synthesized, which is the increased fatty acids levels. And we know increased fatty acids levels in starvation, right? We break down triglyceride and release fatty acids during the starvation. We utilize fatty acids for energies. But under the other pathologic condition, for example, diabetes, the diabetic patients also have very high levels of fatty acids. And I think I mentioned this before, right? Diabetic patients that the lipolysis rate is increased. One of the reasons because of insulin, either insulin deficiency in type 1 diabetes or insulin resistance in type 2 diabetic conditions, the insulin cannot efficiently suppress lipolysis, right? So lipolysis rate is increased, that causing increased fatty acid levels. So fatty acid levels increase also can promote the ketone body synthesis, which is the pathologic condition we're going to talk about later about is so-called ketone acidosis, right? And where actually ketone bodies are synthesized, in which tissues, organs? You guys know liver? I heard liver. Yes, thank you. Liver is the site where ketone bodies are synthesized, right? And particularly in the mitochondria, in the organelle, right? In the mitochondria, liver. So at the bottom part, I just wanted to remind you again the process, for how ketone body is synthesized. So first of all, fatty acids oxidize, right? During the starvation, fatty acids levels increase, or under the pathologic conditions, fatty acids levels increase. Then will a lot of fatty acids be oxidized through the beta oxidation pathways, remember? And generates a lot of acetyl-CoA molecules, right? And acetyl-CoA molecules either can go to the TCA cycles or for the synthesis of ketone bodies, and depends on the tissues, right? For example, in the skeletal muscle, and muscle utilize a lot of fatty acids, so lots of acetyl-CoAs being directly oxidized through the TCA cycles for ATP production, right? But in the liver, because liver is the site, right, for ketone body synthesis, so lots of acetyl-CoA actually go this pathway for ketogenesis in the liver. So again, depend on which tissues and which pathways, the acetyl-CoA molecules will go. And the other interesting thing is that ketone body is synthesized in the liver, but actually not utilized in the liver. Right? So ketone body, once it's synthesized in the liver and released into the circulation and utilized in other tissues, we know that brain is one of the important tissues that utilize ketone bodies, and also other tissues like muscle, heart, and kidney will also utilize the ketone bodies, right? And we're going to explain the reason why liver cannot use the ketone bodies. Okay, so ketone bodies, there are actually three compounds, acetoacetate and the beta-hydroxybutyrate are the two common ketone bodies in the body. And acetone is actually a side product of the ketone bodies. So for example, the acetoacetate can be directly or spontaneously deposition, right, to the acetone. So acetone can be excreted through the lungs, and that will cause a kind of special smell. I don't know if you guys heard about the acetone breath. The smell is very special, like a fruit or kind of nail, polish remover or something like like this kind of smell. So and in the diabetic patient, because of lots of ketone bodies synthesized, right? So you can tell by the diabetic patient's breath, kind of special smell, the fruity smell, the ketone acetosis. And these two molecules, like acetoacetate and the 3-hydroxybutyrate, can actually interconvert these two ketone bodies. And the enzyme is so-called 3-hydroxybutyrate dehydrogenase, can catalyze this interconversion of the two ketone bodies. Right? Any questions about so far? Right? Okay, so here I'm going to talk about the bit detailed the process of a ketogenesis, including what are the starting molecules or substrates of ketogenesis. And you can look at this block, first, right? So ketogenesis, the substrate actually acetyl-CoA. Right? Acetyl-CoA is the starting molecules or substrates of ketone bodies. So ketone body basically is synthesized from acetyl-CoA molecules, right? I think we mentioned in a couple of slides earlier. So two acetyl-CoA molecules can be condensed to acetyl-CoA, right, by this enzyme called silase. And acetyl-CoA, and the next step is acetyl-CoA and together with another molecules of acetyl-CoA, right, being formed the HMG-CoA. HMG-CoA have a long name like 3-hydroxyl 3-methyl glutarate-CoA, right, HMG-CoA molecules. An enzyme catalyzing reaction is the HMG-CoA synthase, right? And then the following step is the breakdown of HMG-CoA molecules by HMG-CoA lyase and release the acetyl-acetate and the acetyl-CoA molecule. And this acetyl-CoA molecule is being recycled, right, and enter the next round of the reactions. So, and these molecules that we already mentioned, like can interconverted to the 3-hydroxybutyrate, right? So several reactions for synthesis of ketone bodies, and again the substrate or initial or precursors of ketone bodies is the acetyl-CoA molecules, right? And this diagram just gives us a little bit more details, the synthesis. And we know that, again, the acetyl-CoA molecules primarily from the beta oxidation of fatty acids, right? So fatty acids and beta oxidation and produce its products, it's acetyl-CoA, right? We all know that. And what about the other sources of acetyl-CoA? Other nutrients can also oxidize, right, and produce acetyl-CoA. What are the two other nutrients that can also produce acetyl-CoA after oxidation? Yeah, pyruvate, but lactate, but more, upstream of glucose and amino acids, right? And why fatty acids is more important than glucose and amino acids in terms of the ketone body synthesis? Thinking about the carbons, right? Fatty acids have more carbons, right? That means they produce more acetyl-CoA molecules compared to glucose and amino acids. So fatty acids is most important sources of acetyl-CoA, right? And for ketone body synthesis. So and I'm not going to go through this, and pretty similar like here that's described, right? First of all, the synthesis or condensation of the acetyl-CoA molecules to acetate-CoA, and then additional acetyl-CoA molecules and form together with the acetyl-CoA form the 3 HMG-CoA molecules and HMG-CoA molecules, and then break down again by HMG-CoA lyase and to acetyl-acetate and release the acetyl-CoA molecules, and then it will go be recycled, right? So for the synthesis of ketone bodies or the process of ketogenesis, what you need to know, just try to understand the substrate of the ketogenesis, which is acetyl-CoA, right? And which sources of the nutrient oxidation that contribute to the acetyl-CoA for ketogenesis, which is primarily the fatty acids, right? And liver cannot utilize the ketone body, right? Synthesize. Again, this whole process occurred in the liver. Here is the sort of whole picture, where the ketone body is synthesized and where ketone body is utilized. So again, the liver is the major or the primary site for ketone body synthesis. Again, fatty acids is a major contributor to the substrate of ketone bodies, acetyl-CoA. And glucose can also contribute, but it's much less, right, compared to the fatty acids. So, and ketone body, acetyl-CoA once is generated, then go to the ketogenesis pathways in the liver. Also have some can go to TC cycle, but majority of acetyl-CoA in the liver goes to the ketone body synthesis pathway during the starvation conditions, right? So in starvation, this pathway be highly activated, right? And once the ketone body synthesized in the liver and released into the circulation, some of the ketone bodies can be excreted through the urine, or the other ketone bodies being converted to acetones and excreted in the lungs. And utilization, lots of other tissues can utilize the ketone bodies, right? I mean, including brain, skeletal muscle, heart, and kidney. So in this, all this so-called exhepatic tissues utilize ketone bodies. So once ketone body enter these tissues, and first of all need to be converted back to acetyl-CoA, right, through the multiple reactions. We're going to talk about it next slide. And acetyl-CoA then enter the TC cycle for ATP production, of course, involving the electron transport chain, right, for ATP production. So this is sort of a whole process of the ketogenesis, as well as the utilization of ketone bodies. Okay, so now let's go to sort of a biochemical process of the ketone body utilization or oxidation of ketone bodies. The ketone body is synthesized in the liver and transported to the other tissues. Here we just as the skeletal muscle as an example. So once enter the skeletal muscle, the two common forms of ketone bodies, like acetoacetate and 3-hydroxybutyrate, can be need to be, first of all, they need to be need to be converted to acetyl-CoA. So one CoA group be added to acetoacetate molecules, right, to form the acetyl-CoA. And the CoA group basically is from succinyl-CoA. Succinyl-CoA, is the intermediate of the TC cycle, right? Remember? So, so the enzyme that carries this reaction is called the succinyl-CoA transferase. So transfer CoA group from succinyl-CoA to acetone and generating acetyl-CoA molecules, right? And the next step will be the breakdown of acetyl-CoA, acetyl-acetyl-CoA to two acetyl-CoA molecules by silase enzymes, right? And acetyl-CoA molecule, once it's formed from the ketone bodies, then enter the TC cycles, right, for oxidation and ATP production. And here I wanted to point out this succinyl-CoA transferase. It's a key enzyme, right, for utilization of ketone bodies. So, and ATP, 2 ATP actually used for this reaction. So now you'll probably be able to answer the question why liver cannot utilize the ketone bodies. Just, guess why. Yes, because it lacks of succinyl-CoA transferase, which is a key enzyme for the utilization of ketone bodies, right? So liver does not have this enzyme, so cannot break down the ketone bodies and to utilize ketone bodies. Right? Any questions? Yes. Oh, your question is this muscle is able to utilize fatty acids, why it's needed to utilize ketone bodies? Well, during the the prolonged starvation, because the glucose neurogenesis goes down and glycogen probably almost depleted, so the lot of tissues need to utilize more fatty acids, and particularly for the brain, at very late stage of starvation, brain also try to utilize the ketone bodies. So that means that the ketone bodies require the requirement excuse me, the demand for ketone bodies is a significant increase during the later stage of starvation, right? So that means the body need to utilize a lot of fatty acids for make ketone bodies, right? So that at that stage, the sources of or availability of fatty acids for muscle energy use also kind of decline. So that's why the muscle is also need to utilize some ketone bodies, right? Now that makes sense? Okay. All right, so here is the energy yield from palmitate. We kind of comparison if palmitate directly complete oxidize versus oxidized through the ketone body synthesis, right? So for example, we know that palmitate, the fatty acids can be oxidized through the beta oxidation in mitochondria and produce the 8 acetyl-CoA molecules, right? Palmitate is, again, it's 16 carbon fatty acids and produce 8 2-carbon unit acetyl-CoA molecules after beta oxidation. And if 8 acetyl-CoA molecules all enter the TCA cycle for ATP production, it can generate, we already calculated, right, about 129 ATPs. But if acetyl-CoA not like directly to the TCA cycle but go to the ketone body synthesis, right? And we know acetoacetate is a 4 carbon molecules, right? So if 8 acetyl-CoA molecules enter the ketone body synthesis pathways, can generate 4 acetoacetate molecules, right? So if 4 acetoacetate ketone bodies being utilized, remember that key enzyme for utilization ketone bodies, succinyl-CoA transferase. This reaction requires 2 ATPs, right, to break down the acetoacetate molecules. So utilize 4, sorry, not 2 ATPs, 1 ATP. I apologize for that mistake. So succinyl-CoA transferase, you need to utilize 1 ATP for reaction, right? So 4 acetoacetate breakdown by succinyl-CoA transferase and utilize 4 ATPs, right, here. And generating 8 acetyl-CoA molecules. And these 8 acetyl-CoA molecules eventually enter TCA cycle for ATP production. Also, it produces 129 ATPs, but because they utilize 4 ATPs, right? So the net production of ATP from this beta oxidation and ketone body utilization pathways and generating 129 ATPs, right? So, they produce a little bit less ATPs compared to directly oxidizing TCA cycles, right? Okay, so now I wanted to talk about the regulation of ketogenesis. We know ketone genesis occurs during the starvation, but how or in what conditions that promotes this process, right? Or why ketogenesis is increased or stimulated during the starvation. So the three main steps that can control the rate of ketogenesis. And first of all, we know that during starvation, lipolysis is increased, right? To break down the triglyceride and release fatty acids. So this is the first and step that can control ketogenesis, right? Fatty acids basically provides oxidize and provides substrate for ketogenesis, right? So the availability of fatty acids is most important. So lipolysis, if anything, any factors that can regulate lipolysis, either increase lipolysis or decrease lipolysis, can control the availability of fatty acids, right? So then can control ketogenesis. So this is the first step, a key step that regulates ketogenesis. And the second step is the blood fatty acids levels is also important. So blood fatty acids levels change not only because of lipolysis, but also some other important factors can also contribute to the fatty acids levels. For example, some kind of disease conditions, fatty acids cannot be oxidized, right? So you also, accumulate in the circulation. So the second factors that circulating levels of fatty acids is also important for ketogenesis, right? Again, similar mechanism, you have more fatty acid, then you oxidize, produce more acetyl-CoA, right? More substrates available. And the third step, the last step, if you only have a fatty acids, it's not enough, right? We need, ketone body needs acetyl-CoA. So fatty acids need to be oxidized to produce acetyl-CoA. And we know that fatty acid oxidation through the beta oxidation, right, occurred in the mitochondria. Remember, we talked about fatty acid oxidation, and fatty acids need to be transported into the mitochondria from cytosol, right? So the key point, a key step that control fatty acid transport into mitochondria, the enzyme we talked about before, right? CPT1. So CPT1 is the key enzyme that control fatty acids transport from mitochondria from the cytosol into mitochondria for beta oxidation and for generation of acetyl-CoA, again, which is a substrate of ketone bodies, right? So try to remember these three key steps that control ketogenesis by control availability of the fatty acids. They control the oxidation of fatty acids, right? Again, this lipolysis, blood levels of fatty acids, and the fatty acid transporter oxidation, right? CPT1. Right. Any questions so far? Right. Now let's talk about some kind of pathological conditions, ketoacidosis, which related to the ketogenesis, right? So ketoacidosis is the pathological metabolic states, and it causes by extreme uncontrolled ketogenesis. Right? So it means that they have very high levels of ketogenic activities, and causing the high concentrations of ketone bodies in the bodies, right? And so ketogenesis is so ketoacidosis can be actually smelled on a person's breath. I just described the acetone, right? It's the specific molecules have a special smell, like fruity smell. So you can smell in person if person has the ketoacidosis. So the most common, so ketoacidosis are the most common in the sort of uncontrolled diabetes or the prolonged alcoholism. So people also describe the ketoacidosis into two different types, like diabetic ketoacidosis or alcoholic ketoacidosis. So here we just want, don't worry about the, this two slides. I'm going to post it, this two slide, right? So here just wanted to talk about the sort of mechanism, and how ketoacidosis that occurs in the diabetes or alcoholism. And ketoacidosis, the main reason is because they have very high levels of ketone bodies, right? Or have lots of ketone bodies synthesized, accumulate in the bodies. So we can go from the bottom to the top to figure out, what could be the reason, why ketone body synthesis increased in the diabetic alcoholism. We know that substrate, right? The ketone body synthesized. So ketone body synthesis requires more, if increased, it requires more substrate acetyl-CoA molecules, right? So acetyl-CoA molecules increase, that will, that will have causing the increased ketogenesis, right? So what conditions that acetyl-CoA production will be increased? In what metabolic conditions or biochemically? Where actually ketone body, acetyl-CoA comes from? Fatty acid oxidation, right? So you will expect fatty acid oxidation will be increased in diabetes or the alcoholisms, right? That will produce a lot of acetyl-CoAs and push, the ketogenesis, right? So you synthesize a lot of ketone bodies. So why fatty acid synthesis of fatty oxidations increase in diabetic or alcoholism? Yes, excellent. Thank you. So because let's thinking about diabetes first. So diabetes, the main problem is that glucose cannot be uptake into the cells, right? Either due to the insulin deficiency in type 1 or insulin resistance in type 2 diabetes. So glucose being accumulated in the circulation to increase blood glucose level, but actually the cells, the inside of cell glucose levels lower, decrease, right? So have a energy crisis, right? The cells, cannot have a lot of glucose available for energy. So that pushes cells to utilize more alternative fuels, fatty acids. So glucose utilization decrease is the main reason that causing increased fatty acids oxidation and increased availability of acetyl-CoA substrate, the ketone bodies. So this is sort of the mechanism. And for alcoholism, because it's causing the dehydration and somehow the blocks the first step of gluconeogenesis. Depleted oxaloacetate. We know oxaloacetate is the important, the mediates, right, of the gluconeogenesis. So alcoholism it actually inhibits or blocks the gluconeogenesis. So glucose cannot be synthesized efficiently in the prolonged alcoholism, right? So causing, decrease the glucose availability and glucose utilization. So from the different mechanism, but the consequence is the same for diabetes and alcoholism. They both have the problem with the glucose utilization. All right, so now I'm going to put the, question, yes.
So you said alcoholism causes decreased gluconeogenesis?
Yes, decreased gluconeogenesis.
So why would there be less glucose?
Well, decreased gluconeogenesis, you cannot synthesize glucose, right?
What about just that you're consuming glucose through food? You wouldn't need gluconeogenesis.
Right, but this is sort of a prolonged, alcoholism, not like just you drink, alcohol just one time, but it's prolonged.
So, but if you have alcoholism and you're consuming glucose, you wouldn't need gluconeogenesis. So what happens with the glucose that you consume? The liver just doesn't break it down.
Okay, so this is the one reason, and the other reason they also interfere with the fatty acids oxidation in the alcoholism. Well, actually, I did not put, the alcoholism is also causing the directly increase the fatty acid oxidation. By the mechanism, it's a little bit complicated mechanism we're going to talk about later, the regulation stuff. So alcoholism, one is, decrease the gluconeogenesis. The other mechanism is to directly increase the fatty acid oxidation, right? Okay, so for the next two slides, I wanted to talk about the regulation. Just go back to the fatty acids oxidation, fatty acid oxidative metabolism, right? And how fatty acid oxidation and being regulated in the fat and fasting conditions, right? So let's look at the so fat condition I highlight in red, right, and fasting in blue. So let's look at the fat condition first and see how fatty acid oxidation being regulated. What do you expect fatty acid oxidation activity in the fat condition? We should upregulate it or downregulate it. Fatty acid oxidation in the fat condition when we have lots of food available and eat a meal. Just guess, suppresses, right? Because we don't need to oxidize fatty acid. We have enough glucose from the diet, right? Glucose is enough, for the energy production. So in the fat condition, fatty acid, in general, fatty acids oxidation should be suppressed, right? Suppressed. So now let's see how fatty acid oxidation is decreased or inhibited in the fat condition. So in the fat condition, we eat the food and we absorb a lot of glucose. We should expect the glucose level will be increased, right? And which causing the increase the insulin levels, right? And insulin is very important hormones that regulates the. Now here we just talk about lipid metabolism, including lipogenesis and fatty acid oxidation, right? This actually the two opposite pathway, right? Lipogenesis basically is for the synthesis of fatty acids, and fatty acid oxidation is oxidized, break down fatty acids, right? So you would expect lipogenesis pathway will be up-regulates, right, in the fat condition. We have lots of glucose, and absorbed, and we oxidize glucose for the energy, but we still probably have excess of glucose when need to to store glucose, right? So as a fat, so the lipogenesis pathway should be also upregulated. So first of all, insulin is upregulated, right, by increased glucose. And the key enzyme that regulates lipogenesis, you remember? ACC, right? Acetyl-CoA carboxylase. And acetyl-CoA carboxylase is regulated by insulin. So insulin basically stimulates the ACC, right? So then drives the lipogenesis to increase lipogenesis activity to convert it excess glucose to fat, right? So this ACC is the key enzyme for this reaction, the formation of marino-CoA from acetyl-CoA, right? So this is the one step. And once ACC activities increase, what you would expect, marino-CoA levels increase, right? You catalyze the formation of marino-CoA from acetyl-CoA. So, and marino-CoA is very important molecules. I would like you guys to remember this molecule, right? It's very important molecules. It's actually very important regulators of CPT1, right? So in general, the marino-CoA inhibits CPT1 activity. Means that when marino-CoA levels increase, that will decrease the CPT1 enzyme activities. And CPT1 is the key enzyme, right? You know that transporter as fatty acyl-CoA into mitochondria for beta oxidation, fatty acid oxidation, right? So now you understand that in the fat condition, the lipogenesis is increased and fatty acid oxidation is decreased, right? And ketone body synthesis is also decreased, right, in the fat condition. So the key regulators is actually the insulin, right? And this key steps as ACC enzymes and the key molecules of marino-CoA, right? So again, in the fat condition, when we have increased glucose and then stimulate insulin release, insulin inhibits insulin stimulates ACC and stimulate the lipogenesis key reactions and produces more marino-CoA. So marino-CoA is the important regulators that suppress the CPT1 and decrease the transport of fatty acids and decrease fatty acid oxidation and ketone body synthesis, right? So this is how, these two lipid metabolism pathways being regulated in the fat condition. Now let's look at in the fasted condition. So again, what you expect, the fatty acid oxidation will be increased, right, in the fast condition, and lipogenesis will be decreased because we wanted to utilize fatty acid, not synthesize fatty acids, right? So, and in the fasted condition, we expect the fatty acids level will be increased because of lipolysis is increased, right? So, and which causing the acyl-CoA, long-chain acyl-CoA molecules levels increase. Remember how long-chain acyl-CoA molecules regulate this ACC enzymes? Remember? Decrease, right? So decrease the ACC. And meantime, because glucagon levels also increase during the fasting, and glucagon is also the regulator of ACC. So two factors together, suppresses the ACC activities, so decreases lipogenesis, right, activities. And once this ACC activity is reduced, then marino-CoA production is also reduced, right? and lower myelin-CoA levels, you have a higher CPT1 activities, right? So and you transport more fatty acids into the mitochondria, so increase oxidation and ketone body synthesis, right? Is that clear? Okay, so try to, thinking about how in different physiological conditions, these two different pathways, lipogenesis and fatty acid oxidation, being regulated. So, and that involve multiple factors, right? And the key will be the hormones, insulin, glucagon, and also the key intermediate molecules like long-chain acyl-CoA and myelin-CoA, right? And key point would be, steps would be ACC catalyze the first reaction of lipogenesis and the CPT1 that control the transport of fatty acids into the mitochondria, right? Any questions? Okay, so I guess this is all for the fatty acids oxidation and ketogenesis, right? I think we probably will take about a 10 minutes break and we'll start a quiz at 11:45, right?